So what I'll tell about today is about our project modeling the tannocide barrier, a gateway for obesity drugs to the brain, to give it a sharp angle here. And yeah, just briefly about myself and my background. I'm professor at the Department of Pharmacy here at the University of Copenhagen. And our group have been working with barrier models for a long time, barrier models in the body. Basically, we have been working with intestinal barriers, lung barriers, kidney barriers. Then we started to work on the blood-brain barrier, and we come from the Department of Pharmacy, so our overall goal is to investigate transport of drug compounds from the blood to the site of action, to the target. So for the last 10 years, we've mainly been focusing on the blood-brain barrier, and we have been making models in the laboratory, in vitro models of this strange and peculiar barrier, the blood-brain barrier, which blocks uptake of drugs from the blood to the brain targets. And I can also add that, do you see my arrow here on the screen?
Yes, yes.
A lot of these studies are made on very simple setups. We try to reduce these complex tissues, and then we have very simple, small, like cups. It's almost like coffee filters in the bottom of a cup, a permeable support where we try to grow the barrier cells in an environment where we mimic all the surrounding media and perhaps also other cell types, but try to get a fairly simple barrier where we can investigate transport. Then when we look at brain uptake of drug compounds, we normally work with the blood-brain barrier, which is simply the capillary wall of the capillaries, the smallest parts of the vasculature in the brain. And here you see a schematic of these capillaries entering the brain. You see this drawing here on the right-hand side, and you can see the red is capillaries going into the brain, and where BBB is written, it's the so-called blood-brain barrier, the very, very tight capillaries of the brain vasculature. If you can cross these capillaries, you can hit almost anywhere in the brain, but they're also really tight because we want to protect the brain, so they keep most drugs out. But also in the brain, there are regions we call the CVOs, or this stands for circumventricular organs, where we actually have leaky barriers, where compounds can leak out. Traditionally, in drug delivery, we have been stating that these areas are so small that they really don't matter, because the surface area is so small. This may be a bit strange thing, because that's actually where we have leaky barriers where compounds can enter. But they enter in very specialized areas. The circumventricular organs means organs close to the ventricles, to these sacs filled with liquid, with CSF in the brain. And they have a very specialized role there. So if we look a bit, we started in a project together with people looking at the uptake of peptide hormones and their effect in the hypothalamus. And they actually had this idea that the blood-brain barrier was not the important thing, but more these circumventricular organs. And we sort of agreed on that because we had investigated transport of insulin, for example, across the blood-brain barrier, and saw, almost no insulin transport. But what we also saw in literature was huge accumulation of radiolabeled insulin in the circumventricular organs. And if we look a bit on the barrier here in this slide, to the left you see the blood-brain barrier, where in the inner part you have the capillary, then these really, really tight endothelial cells. They're surrounded by other cell types, like pericytes and astrocytes. Neurons are in this drawing as well. They're not actually in the real blood-brain barrier, but that's not important here. Then when you look in the circumventricular organs, and especially near the hypothalamus an area called the median eminence. There you'll actually have these capillaries. You'll have the bloodstream, but there the barrier is leaky. You have other cells, but then you have some very specialized cells called tanycytes, which are very long cells, which goes close to the capillaries, and then they go up and make another barrier here before they enter the brain tissue. And this strange barrier, the circumventricular barrier, there in this area, it's not the endothelial cells which makes a barrier. It would be these cells, strange cells called tanycytes. And they're quite mysterious cells. They probably have a lot of roles. Well, in a project, well, I better start with the beginning. If we look at this rather complex area called, this would be data from mice from a publication from 2014 by Langlet. But you have this area called the median eminence. You have capillaries here. These capillaries are leaky, so we are in the circumventricular organ. And then you have these tanycytes sitting. They actually have a barrier up here. They have a tight junction zone, so they're glued together. Then they have another part of this area called the arcuate nucleus, where you have a lot of neurons important in metabolic regulation. There you also have tanycytes going in, but only in this area called the median eminence, you'll have compounds leaking out of the blood. Which means, and I'll skip all the details here, which means that tanycytes actually may be mediators of peptide hormones by transport from the blood to other tanycytes with receptors and to the arcuate nucleus. If we go to the next slide. So in a study basically on liraglutide by our good friend and collaborator and part of the applicant group, Vincent Prévost, it was actually shown that liraglutide was taken up from the bloodstream into the ventricles and further to the arcuate nucleus, most likely by GLP-1 receptors. And this process could actually also be inhibited by various processes which stopped the transcytosis process. So in this study, we were a minor part of the study, but in our group, we contributed a bit with making an in vitro model of this tanycyte barrier from the blood and into the arcuate nucleus. And in brief, to make a very long story short, we took tissue and cells from tanycytes from this median eminence. They were plated on these permeable supports. They were able to grow. We could see they were labeled with a marker, antibodies, and we saw expression of GLP-1 receptors in this area. These tanycytes, they made tight junctions. This rather blurry image would be cells and tight junctional proteins around the edges. They also showed a protein called Z01, which is also a tight junction protein in this upper part, in the apical part, showing they made a barrier. It was not a super tight barrier, so it was not easy to investigate transport across this lab-grown tanycyte barrier. But when we looked at uptake studies, we could see that liraglutide, if we added it to endothelial cells of the blood-brain barrier, a radio-labeled compound, and then tried to inhibit this out with unlabeled compound, we did not see a significant uptake in blood-brain barrier endothelial cells, but in these tanycyte models, we saw a large uptake of liraglutide, and it could be inhibited. We also saw cellular accumulation, and when we sort of filled the cells with liraglutide, and then when we stopped this, it was coming out again very fast. This model was not tight, so we could not distinguish transport from the two sides. But we could see the receptors were present. We got accumulation and release of liraglutide, and we had the presence of GLP-1 receptors. So the project idea that we have and we propose here is it's part of a larger collaboration we have in the applicant group. But we would very much like to generate an in vitro cell model of the tennesside barrier. A model which is tight enough to demonstrate vectorial transport from the blood side to the brain side. A model where we can investigate this transcytosis, where we have the receptors relevant in the median eminence. And this would enable us both, of course, to look at peptide hormones and how they cross from the bloodstream to the brain, but also to screen new medicines, new APIs, new compounds before they are tested in animals and humans. And the work we propose include an in vivo characterization of the receptors at the tennesside barrier. There are made some investigations here, but we would like to go in detail with this. We would like to work on an in vitro setup, a culture setup of tennessides. Ideally for simple purposes, if we could make a model like we already have just with a simple monolayer of tennessides, that would be perfect, but we would probably need to go deeper to go for more advanced 2 or 3D models with more cell types than the tennessides in order to get the correct signaling from the cell environment to get the phenotype of the tennessides. We would like to validate the tennesside model for screening purposes. So we would like to investigate in vitro whether the processes we can see in vivo are all present. And therefore we have a check group working a lot with peptides, which works in the hypothalamus and are capable of also producing new peptides and radio-labeled and fluorescent peptides. And we would also very much like to investigate the transcytosis processes and the rate-limiting steps in the uptake of compounds across tennessides. So if we go to what will we use there of tools, just to mention again that in my group, we have a lot of tools available from our previous work with modeling the blood-brain barrier, the brain capillary endothelium. Basically, we normally start with using these so-called transwells, which are the porous membranes where cells can grow. They're divided, they divide two media, one with blood composition, one with brain composition. And we can add the barrier cell line alone. We can have other cell lines which release soluble factors. We can have cells which can reach our barrier cell in question by sending protrusions through the permeable supports here, and we can even have 3 types of cell, 3 cell types in the same barrier model. We also have the possibility of going for hydrogel models, which are a bit more complex because we see then we would have hydrogels on these supports, but this will slow down processes, especially of larger molecules, a lot. Yes, the unmet need we think there is, but this we would also like feedback on, of course, would be a sort of a fast screening model for brain permeation of novel. And here we say weight-reducing drugs because, yeah, you obviously know the story of Wegovy and the success of Novo Nordisk, Eli Lilly, and so on. But there are probably wider perspectives than this because at the moment the tennessides and all the processes, the neuroendocrine signaling also seems to be involved in a neurodegenerative disease. So drug compounds targeting these neuroendocrine pathways in the hypothalamus are likely to have a much broader aspect than just metabolism regulation. We would like more, and hopefully also the industry, more knowledge on rate limiting steps for CNS peptide drug compounds. This will give us a mechanistic knowledge on drug uptake transport processes, which we also believe and hope is of value for the industry. And we would also get less animal experiments, because we would be able to do experiments just with animal tissue. Of course, we would still need to sacrifice animals, but these could also be animals used for other purposes. So there might be an angle there in 3R settings. We will actually reduce use of animal experiments as such. The academic applicant group, I briefly mentioned the persons and the roles. We would have the Morten Schalborg Nielsen group from Aarhus University working on transcytosis and imaging, being able to investigate on the individual tannosites how compounds are moving. We would have the Vincent Prévost group from University of Lille in France, a tannosite expert, an expert in the neuroendocrine signaling in the hypothalamus. My group, experts in vitro barrier modeling, and the Lenka Malentinsky group from Prague, basically experts in compounds and also able to generate a lot of fluorescent compounds and tools for us. And we believe this group is capable of trying to answering the academic part of the project. But we are, of course, also looking a lot for industry partners and input on the needs, because at the moment, this is still our idea, and we are sort of flexible at this stage, of course. So we would very much like feedback on the project idea. We would very much like feedback on industry needs. We would very much like data sharing on relevant topics, and basically also participation. And that was all from me for the talking part. Then I would be happy if there are questions or comments for discussion.